Assalamualaikum warahmatullahi wabarakatuh. I bid to Dr. Hamzah bin Muhammad Nasir, our respected lecturer on subject SBT 3313 Immunology, and fellow friends. I am Rahil Binti from Group 1. We present to you about our problem-based learning mini report on case study 1, which is beta-thalassemia major. The case study problem statement stated that Paul, which is a 7-year-old Asian male, has been diagnosed to have red blood cell defect, which is beta-thalassemia major. He was consulted to a medical specialist and was proposed for a bone marrow transplantation. He has no sibling, however, his mother has subscribed to a cryopreservation of cord blood. Hence, as a medical biotechnologist, our group will review this case and propose a solution with respect to immunological discipline. Kuliyyah anemia, or scientifically known as beta-thalassemia major, is categorized as one of genetic blood disorder which is inherited from defective or mutated beta-globin gene of both paternal and maternal that is passed down to their progeny. This beta-thalassemia major was first described in the year 1925 by Dr. Thomas Benton Coley, which is an American pediatrician. In this particular type of thalassemia, the progeny will inherit the most severe type of anemia, which is a condition where red blood cell or RBCs is insufficient or abnormal. This condition will cause the iron on the RBCs to unable to carry out the function of transporting the oxygen throughout the body, thus affecting the natural body mechanism. The real cause of beta-thalassemia major is originated from lack or absence of beta-globin chain in the synthesis of normal alpha to beta-2 hemoglobin tetramer. This will subsequently lead to the ineffective degradation process of alpha-globin chain, affecting erythropoiesis, which is the production of matured RBCs from erythropoietic stem cell, causing hemolysis to happen intensely and further lead to anemia. Anemia will also cause the synthesis of erythropoietin or EPO, which is the hormone produced in kidney, to be disrupted. All this effect will contribute to beta-thalassemia symptom, which will be experienced by beta-thalassemia major patient in such a young age, which in this case refer to Paul.
So, what are the immunological requirements for bone marrow transplantation? There are two types of bone marrow transplantation, which are autologous bone marrow transplantation and allogenic bone marrow transplantations. An allogenic bone marrow transplantation requires 3 immunological conditions in order to be able to be transferred to a patient. The first one is the compatibility of human leukocyte antigens, HLA, precisely MHC class II antigen of the donor, whether they are from siblings or matched unrelated donors. This requirement is the most important out of all. Siblings' bone marrows are the nearest match to the patient, having higher graft survivability compared to unrelated match or mismatched donors. The second requirement is the ABO blood type compatibility of the donor and the patient. A mismatch in blood type can cause transplant rejection, which is similar to blood transfusion reaction. However, it can be treated by taking immunosuppressive therapies or drugs. And lastly, immunosuppressive preparations and therapies are required for allogenic bone marrow transplantation. The transplant needs to be pre-treated by using cytotoxic and myeloablative agents in order to avoid graft rejection. On the other hand, autologous bone marrow transplantation can be carried out after chemotherapy or lethal irradiation and treated with antibiotics after the transplantation occurs.
To help in this case, an autologous genetically modified hematopoietic stem cells transplantation by biotechnology techniques such as gene therapy and gene editing can be performed as treatments for beta-thalassemia. Gene therapy is a viral vector-based therapy in which living drug is used to fix errors in the defective genes. On May 2019, Bluebird Bio, a biotechnology company, has produced the latest gene therapy product that called as Zynteglo. Zynteglo utilizes the lentiviral vector to carry the normal beta-globin genes and to be transduced with the DNA of the CD34 plus cells, which is the progenitor stem cells derived from the bone marrow. Once the cell integrated with the correct beta-globin DNA, they will be able to proliferate into normal blood cells. The genetically modified stem cells will be infused back to the patient's body and replace the mutated stem cells in the bone marrow. It was reported that 4 out of 5 patients with non-beta genotypes are successfully been transfusion-free after more than 12 months of follow-up sessions. Besides, gene editing is an approach to correct genetic mutations of DNA specifically by utilizing targeted nucleases to restore the wild-type sequence of the gene. One of the current gene editing therapy in development for beta-thalassemia patients is CTX100. In this method, the aim is to reactivate the fetal hemoglobin that consists of 2 alpha and 2 gamma chains to adult hemoglobin that has normal 2 alpha and 2 beta chains. It was observed that fetal hemoglobin has a greater affinity towards oxygen and being functional after being reactivated. This can be achieved by the description of the key transcription factors of the gene such as BCL11A gene that are responsible for the switching of gamma to beta chains. To inhibit the gene expression, the genomic editing tool CRISPR-Cas9 has been used to perform deletion on erythroid-specific enhancer of the BCL11A gene. This study is supported by the clinical report from preparation and method with CTX100 has successfully become transfusion independent after 12 months of treatment.
Okay, so now I will explain to you the general clinical procedure for beta-thalassemia patient that will undergo autologous hematopoietic stem cell transplantation. Okay, so first, the patient will be injected with certain drugs to induce stem cell mobilization that will increase the number of hematopoietic CD34 plus cell migration from the bone marrow to the peripheral blood vessel, which then will be withdrawn from the patient. So the CD34 plus cell or the stem cell will undergo gene modification, either gene therapy or gene editing, which has been explained by Qiestiena before, and then they will be cultured until adequate number of gene-corrected CD34 plus cell achieved. Okay, so before the transfusion of the gene-corrected stem cell can happen, the patient will undergo a treatment called conditioning, where it's basically a chemotherapy session that will suppress the bone marrow in order to give space for the new gene-corrected stem cell. And then the stem cell will be infused back into the patient, and engraftment will occur when the stem cell find their place in the bone marrow. Okay, so for post-treatment, the patient basically need to do follow-up session with their respective transplant physician in order to monitor the efficiency of the treatment. So this autologous hematopoietic stem cell transplantation is basically a lifelong treatment where the genetically modified stem cell will continue to live inside the bone marrow to produce a healthy blood cell so that the patient won't be dependent to the transfusion method in the future.
What are the risk factor and chances of success of autologous hematopoietic stem cell transplant? In general, bone marrow transplant comes with a risk of failure, mainly due to the graft versus host disease and graft rejection. Graft versus host disease and graft rejection depends on the human leukocyte antigen compatibility between the donor and the recipient. However, in this case, the patient pool will be treated by autologous hematopoietic stem cell transplant. The risk factor of autologous stem cell transplant include bone marrow suppression, liver damage, interstitial pneumonia syndrome, infections, and long-term side effects such as cataract, infertility, and new cancers. The chances of success of autologous stem cell transplant are greater than the allogenic stem cell transplant due to the 100% match of human leukocyte antigen, lower risk of graft versus host disease, lower risk of graft rejection, lower risk of viral infection transmission. Last but not least, there is no need to find a matched donor. Several aspects of gene therapy for thalassemia faces several difficulties. These difficulties are related to the vector design, which is very crucial for the efficient translation of hematopoietic stem cells. Another is the return back of non-transduced hematopoietic stem cell to the patient's hematopoietic compartment. Last one is the unknown dynamics of long-term survival of transduced hematopoietic stem cell in patient body due to the absence of donor's immune system.
In conclusion, modified gene of hematopoietic stem cells, precisely cord blood transplantation, has been proposed as the best solution in treating Paul who suffered beta-thalassemia major. This method proved to be effective as it did solve the issue of graft rejection and GVHD since it came from Paul's own cells rather than using unrelated donor's bone marrow since he doesn't have any siblings. Having the cells undergo gene editing to correct the impaired CD34 positive and infused back to Paul's peripheral blood and later into Paul's bone marrow will induce the production of normal red blood cells for Paul. This is only a proposed solution to Paul's family after thorough discussions are made before any actions are further conducted. That is all from us. Thank you.